This silent workshop presentation is about how you can increase your laboratory efficiency with thermoscientific biplates. But first, I would like to ask you, do you need to be more efficient in your lab and to increase your productivity, and at the same time be more environmentally friendly, ergonomic with a work-friendly environment, deliver accurate results with high quality, and on top of that, achieve all these goals with less resources, sourcing savings, and more regulatory requirements? We can offer you the solution. You can be more efficient with less material in use by processing the same amount of samples. How is this possible? By using biplates. What does this mean? By using a biplate, you have the hands-on time and double your outcome. You can perform 2 tests with 1 plate. This means that you can save space in incubators, so you need less incubators. You have less waste using less plates. You need less hands-on time, also less confirmation tests, and also less additional equipment if you're, for example, using materials for anaerobic atmosphere. And you can increase 50% in output on lab automation. The last point is probably the most interesting one, especially when you're a lab automation user. On this, I would like to present a case study. This case study was performed in UK at St. George's Hospital in London. They have a large state-of-art microbiology lab using 2 Kistra systems. They have approached us to develop a biplate to screen for group B streptococci and gram-positive screening to improve and enhance efficiency and also being more patient-focused. We were able to offer our Brilliance GBS agar, which is a chromogenic media for group B streps, and the traditional CNA medium for gram-positive organism on a single plate. By using this biplate combination, you have a highly selective medium for group Bs, and combining this with the traditional CNA, the detection is not limited to GBS. Gram-positive organisms can also be identified, giving laboratories a more complete clinical picture from a single sample. One main comment was that automation is the way forward. However, automation must be complemented with effective use of process which improve efficiency and quality further. Further comment was also that with the biplate, it's a more selective media for group Bs and other gram-positive organisms, and it allows to identify group Bs rapidly. It is definitely sensitive and it's beneficial to the scientists reading the digital images. And one main comment as well is that instead of having 2 plates, a CNA and a group B strep plate, we have half. So we are producing half of the waste, half the storage, resulting in significant savings in storage, wastage, and cost. And on top of that, the CNA side also allows to pick up staph aureus and toxic shock patients. So that's quite good, and it cuts out all the gram-negative flora, which is massive on a genital swab. Prior to using the biplate, the lab was using CNA monoplate, which required more confirmation tests on multitoph since it was more difficult to tell the difference between group B and group D streptococci. So the new biplate enables them to limit their use of multitoph to just high-risk patient population, such as pregnant women, saving time and cost. This case study shows very well how you can improve your lab efficiency and also supply better patient care. Another good biplate example is the Brilliance Aspel Brilliance CRE agar. This is a biplate to screen for gram-negative organisms, so for detection of multi-drug resistant Enterobacteriaceae, Acinetobacter, and Pseudomonas. By the simultaneous use of both media, this increases the chance of detecting clinically significant OXA48 strains, since these strains are not always detected on a single CRE medium. You can also detect resistant Acinetobacter and Pseudomonas strains. And along with the incubation time of 18 to 24 hours, the Brilliance Aspel Brilliance CRE agar offers efficient and rapid detection of multi-drug resistant gram-organisms. And also the biplate benefits, which was mentioned before, like saving space and automation, incubators, less waste, and less hands-on time. Other biplate combinations we have in our range are for example, traditional media, Columbia agar with endo, or Columbia with meconchime, or Columbia CNA with the Brilliance UTI agar, which is for screening for pathogens for urinary tract infections, or Columbia CNA with meconchime number 3. We have also biplates where you have 2 medium in one plate, which are the same formulation, like the Kamali. In this case, you will save more cost on the materials to producing the CO2 atmosphere for Campylobacter, and also a biplate with SS agar and XLD medium for Salmonella and Shigella detection. Other examples we have is for Brilliance Candida and Saburo GC agar. So Brilliance Candida is a chromogenic media to screen for Candida species. For anaerobes, we have the Schädler anaerobe agar and Schädler kanamycin vancomycin. Here it's the same example as with the Campylobacter media. You can save more costs on the materials to generate the anaerobic atmosphere. And we have the Brilliance Salmonella agar and XLD agar, which is a medium for detection of Salmonella. And with the chromogenic side, you have a very selective media to screen for Salmonella. This could be interesting, especially for customers testing food samples, since the first media to use is the XLD media and the second media is a choice. So this biplate combination offers a very good example. Other media we have in the range is Brilliance MRSA2 agar and Columbia agar with sheep blood plus. Here you have a chromogenic media with a general media. On the Brilliance MRSA side, you can detect presumptive for MRSA, and on the Columbia side, you have a media where you can detect all organisms, and you can also quantify the number of organisms on the sample, so that's a double check that you have organisms on the swab as well. Then we have the clad medium and meconchia agar number 3 and Columbia agar with chocolate agar. Here you can see all the selections we have in our range. I would like to highlight especially the Brilliance UTI biplate, which is interesting, especially for lab automation users, since this can save space in the automation, and since the workflow in the microbiology lab for urinary tract infection samples is very high, this could benefit in cost saving and an increase in the output. If you have any other demands for any other biplate combination, we are very flexible to offer this and happy to help out more. Thank you for your attention, and please contact the booth staff for more information on Thermo Scientific biplates and also the case study which has been presented here.